Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad Integrated MSc PhD Entrance Examination for the subject Biotechnology conducted in the year 2015. I will be discussing only the biology part. So first question. Nuclear sizes are expressed in unit called as Fermi. Answer is option A. Sequence coverage in the whole genome sequencing is dependent on. It is dependent on read length, number of reads, genome size, all of the above. Answer is all of the above. Which of the following can be used to determine an alpha helical conformation of protein in a solution? So circular dichorism can be used. Answer is option B. Then the base adenine of a DNA of an organism is 21.3% of its total DNA composition. Which of the following statement is correct for the base composition of the genome given C is cytosine, G is guanine and T is thymine? So A and T, they will be equal to 21.3% because adenine will be equal to thymine because adenine base pairs with thymine. So it will be 21.3% and GC content, it will be 21.3 plus 21.3, you will get 42.6. 100 minus 42.6 will give you 57.4. That will be the GC content. So G will be 28.7 and C will be 28.7. So the answer will be option A, that is A equal T is equal to 21.3. G equal to C equal 28.7. Which of the following technique is most suitable for detecting the presence of a gene product, that is protein? Gene product is protein. So Western blotting is the most suitable technique. Then a vector able to replicate into two different hosts is called shuttle vector. Answer is option D. An amino acid that has a secondary amine and disrupts alpha helix formation is proline. Answer is option D. The following is a Y-linked phenomenon. It is Hollandric inheritance. Answer is option A. 2,4-D is a structural analog of. It is structural analog of oxine. Answer is option A. Then, which of the following is true? Epigenetic means alteration of both genotype and phenotype upon environmental change. So epigenetics does not involve change in DNA sequence. So that is true. Option B is correct. Then which of the following enzyme uses RNA as template to make DNA? So it is telomerase that uses RNA as template to make DNA. Then reverse transcriptase enzyme is involved in. So it is involved in synthesis of complementary DNA from mRNA. Answer is option A. The oxidized form of glutathione is represented as GSSG. Then next one. You engineered a bacteriophage that had the protein coat of T4 phage and the DNA of T2 phage. The composite bacteriophage was allowed to infect bacteria. What would be the composition of the new phages produced in the bacterial cell? So only foreign DNA is introduced into the host cell genome. Okay, so when bacteriophage infects a bacterial cell, their normal mode of reproduction is to harness the replication, transcriptional and translational machinery of the host bacterial cell to make numerous virions or complete virus particles, including the viral DNA or RNA and protein coat. So when bacteriophage infects, only the DNA enters the cell. So here only DNA of the T2 phage entered the cell. And it enters the cell, but the protein coat does not enter. So T4 phage protein coat does not enter the bacterial cell. So the new phage will have the protein coat and the DNA of the T2 phage. So the answer here is option C. Then what is the chemical basis of gene imprinting? So it is methylation of DNA. This is a repeated question. Then which of the following E. coli sigma subunit is required for the transcription of heat inducible genes? So it is sigma 32. Answer is option B. The substance that is the general biosynthetic precursor of sex hormones and hormones of adrenal cortex. It is cholesterol. Answer is option D. Anaplerotic reaction involves the conversion of pyruvate to oxaloacetate. Answer is option C. Molecular chaperones are, they help protein to fold correctly and repair incorrectly folded proteins. Answer is option D. Classification of bacteria into gram-positive and gram-negative categories is based on staining. Then, with reference to the bacterial cell wall, which of the following statement is correct? Both gram-positive and gram-negative bacteria possesses peptidoglycan, but gram-positive bacteria lack outer membrane. Answer is option D. An example of peptide hormone, that is histamine. Answer is option B. A hydrocarbon is said to be saturated if each internal carbon atom is covalently bonded to two hydrogen atoms. Answer is option C. Then in enzyme kinetics, Kcat by Km indicates efficiency of an enzyme. Answer is B, 2 only. Then rapid but non-antigen specific immune response are produced by the innate immune system. Answer is option B. LYS2 is a selectable marker for yeast. Option C. The size of virus will be maximum in micrometers. So virus range in size from 5 to 300 nanometers. Giant viruses including Pandora virus with a diameter of 0.4 micrometer have also been identified. So the size of the virus will be maximum in the case of is micrometers. Then, which of the following are true with regard to interferons? This is also a repeated question. Activates B cell to make virus-specific antibodies. Which of the following disease is not an autoimmune disease? Type 1 diabetes, celiac disease, Alzheimer's disease, Hashimoto's thyroiditis. So all are autoimmune diseases. I don't know what was given in your answer key. I don't have the answer key with me. Okay. Then, mitosis produces a different number of cells than meiosis. Which of the following statement is true? Mitosis produces 2 cells while meiosis produces 4. Answer is option A. Then, Down syndrome result from the presence of an extra chromosome in position 21. Answer is option B. Which of the following is not the part of a virus? So, plastid is not a part of the virus, it is for chloroplast. Then alternative oxidase pathway is associated with. So alternative oxidase is one of the terminal oxidases of the plant mitochondrial electron transport chain. So the answer here is mitochondrial electron transport chain. Then next one, antigen presenting cells that activate helper T cells must express which one of the following on their surfaces. So it is class 2 MHC antigens. Answer is option D. Which of the following organism would not undergo meiosis? So bacteria doesn't undergo meiosis. Answer is option A. Which of the following genetic elements in bacteria are required from an external donor? So the answer here is option C, that is plasmids. Next one. Which of the following can digest protein? So trypsin and pepsin, they can digest proteins. Answer is option B. Then restriction enzymes which cut the DNA nucleus were discovered by. So they were discovered by Nathan Arbor and Smith. Answer is option D. A light receptor in certain bacteria resembles that found in the eye of animals. What is the bacterial light receptor called? This is also a repeated question. It is bacteriorhodopsin. Answer is option C. Polynucleotide phosphorylase (PNPase) polymerizes following substrates when they are available in excess. So it is DNDPs, dinucleoside diphosphates, DNDPs. An origin of replication. A particular sequence in eukaryotic or prokaryotic DNA at which replication starts. Answer is option A. Then which of the following enzyme was first isolated in pure crystalline form? This is also a repeated question. It was in UoH, not only for integrated MSc entrance but also for other MSc entrance, that is for biotechnology, plant biotechnology, animal biotechnology, biochemistry, molecular microbiology. In one of these question papers, I have seen the same question being repeated. Then, which of the following amino acids absorb maximum UV light at 280 nanometers? It is tryptophan and tyrosine. Answer is option B. The Maxim-Gilbert method of nucleotide sequencing is based on. It is based on chemical modification of DNA. This is also a repeated question. Which of the following is used for global sequence alignment? So it is Needleman-Wunsch algorithm that is used. Answer is option A. Then unidirectional transmission of nerve impulse through nerve fiber is due to the fact that neurotransmitters are released by the axon endings and not by dendrites. Answer is option D. Which of the following is a DNA binding protein? So NF-kappa, that is nuclear factor kappa, is a ubiquitous transcription factor that is involved in inflammatory and immune responses as well as in the regulation of expression of many other genes related to cell survival, proliferation and differentiation. So which is in DNA, one of the following is a DNA binding protein is NF-kappa B. Then the primary thermodynamic factor that serves the formation of a lipid bilayer in aqueous surrounding is, it is hydrophobic force. Answer is option B. The enzyme which converts palmitic acid to palmitoleic acid is, it is desaturase. Answer is option B. Palmitoleic acid is a product of sterile CoA desaturase, an enzyme that converts palmitic acid into palmitoleic acid in the liver. So answer is option B, desaturase. Then next one, gel electrophoresis is to size separate DNA molecules. Answer is option A. Then raw honey is more wholesome than processed honey because it contains honeybees probiotic bacteria and pollen. Answer is option A. So that's it. Thank you for watching my channel. Please like, share and subscribe. Thank you.